Hello, listeners. Yamina here. Welcome to this special episode of the Dr. GPCR podcast. My guest is Dr. Aaron Sato. He is the Chief Scientific Officer and VP of Protein Engineering at Twist Biopharma. Twist Biopharma is actually a vertical of Twist Bioscience. If you've never heard of Twist Bioscience, it created a revolutionary silicon-based DNA synthesis platform to offer customers access to more affordable, high-quality synthetic DNA faster. Aaron, my guest today, earned his PhD at MIT, where he worked on the structure-function relationships of MHC class II proteins. He worked in a variety of roles after his PhD, where he used his protein engineering background and became what I would call an antibody engineering expert. As such, Aaron has been a Senior Director of Lead Discovery, Senior Director of Antibody Discovery, VP of Research, VP of Protein Sciences, and before joining Twist Biopharma and Twist Bioscience, he was the CSO and head of Lake Pharma Antibody Center. Aaron and I talked about how using Twist Bioscience's proprietary technology to manufacture DNA at a scale, the team saw an opportunity to tackle the challenge of identifying novel functional antibodies targeting GPCRs as part of Twist antibody library design. To learn more about Twist Biopharma or Twist Bioscience, please visit twistbiopharma.com or twistbioscience.com. Before we jump into this episode, I wanted to thank Twist Biopharma for sponsoring this podcast episode and our Dr. GPCR October newsletter. If you haven't done so yet, visit drgpcr.com/newsletter to subscribe and share our newsletter with your colleagues. To support Dr. GPCR, please visit drgpcr.com/sponsors or email us at hello@drgpcr.com. And now, let's dive into our episode with Dr. Aaron Sato. So hello, everybody. I'm Yamina Bershish, founder of Dr. GPCR, and welcome to another episode of the Dr. GPCR podcast. Today, I have the pleasure and the honor to talk to Dr. Aaron Sato. He is the CSO currently at Twist Biopharma. Hi, Aaron.
Hi, Yamina. How are you?
Good, thank you. Thank you for being here today.
Oh, my pleasure.
So as I mentioned a minute ago, so you're the CSO at Twist Biopharma. Can you tell us a little bit more about your career and how you got to where you are today?
Sure. So I'll go all the way back to my graduate work. I graduated from MIT in the lab of Lawrence Stern, who's a crystallographer in the area of working on MHC class II crystal structures. So I got a really firm, great experience and working with looking at structure-function relationships around immune receptors. I then transitioned into my very first job, which was at DIAX, which is a synthetic antibody phage display company. So I got a really broad experience on discovering antibodies using phage display. And then I went on to a number of other smaller biotech companies as I transitioned to California. So I worked for Oncomed. It's a cancer stem cell company focused on using biologics to address cancer stem cells. Worked at Sutro Biopharma, where they had a unique cell-free platform for making site-specific antibody drug conjugates. And then I transitioned to a couple other roles. I led the Antibody Center at Lake Pharma, which is one of the largest biologics CROs in the United States. And then finally, I landed at Twist, where I head up the Twist Biopharma vertical at Twist. So that group is focused on utilizing all the fantastic DNA products of Twist to help pharma and biotech companies discover as well as optimize antibodies. And I've been at Twist for 2 years now.
Wow, fantastic! So a bunch of roles that all prepared you at the end of the day to assume this new role at Twist.
Yeah, I mean, I always say that my career has always been leading up to this fantastic role that I have at Twist now, which is basically utilizing synthetic DNA libraries and utilizing phage display to discover antibodies against all kinds of different targets. So it was really a fantastic and perfect role for me to step into.
Maybe my audience is not aware of who Twist Biopharma is, or not even aware who Twist is. Can you tell us more about the difference between Twist Biopharma and Twist Bioscience, and what do you do?
Sure. So again, Twist Biopharma is a division within Twist Bioscience. Twist Bioscience as a parent company has a very unique silicon platform where they can basically synthesize large pools of individual oligos up to 300 base pairs in length. And they can use those pools of oligos to make all kinds of different DNA products. So clonal genes is one of our products. So you can go online, order any DNA up to 5 kb, cloned into any vector you wish. And that's a really large revenue generating business for us. We have other products in the NGS space where we're using pools of oligos for sample prep ahead of NGS sequencing. And then we've created 2 additional verticals in the company, Twist Biopharma being one of them, which I'll expand on in a second. And then the other is data storage. So I oversee the Twist Biopharma division of the company. And that group is focused on, as I said, using another product I didn't mention, which is our DNA library technology, where we can actually use these pools of oligos to make high quality and diverse DNA libraries. We use that same platform to create a whole suite of different antibody phage display libraries. And that's key and central to our mission, which is basically creating those libraries, broadly licensing them outward to other biotech and pharma companies, as well as doing funded research projects on behalf of those companies to help them discover as well as optimize antibodies. And we have a whole platform for doing antibody optimization as well. That's called TAO, which stands for Twist Antibody Optimization. And that again utilizes that same library platform to help others take an initial antibody sequence and improve upon it and potentially also humanize it as we do that.
Wow. I did look around on the Twist Biopharma and Twist Bioscience websites, and I think the technology of being able to print DNA at such a high yield and have all these libraries, it's really revolutionary, and it's really cool technology.
Yeah, I mean, one differential that that's been really great for me as an antibody engineer is that in the past, when we've made synthetic libraries, we've typically had to use degenerate oligos to do that. So either oligos that use mixtures of nucleotides or trinucleotides to encode for the diversities that you see in the CDR loops of an antibody. Using our silicon-based platform, we don't actually have to do that. We can actually make pools of individual discrete sequences and oligo pools, and then use that as the input to then create these libraries. And that gives us a lot more control, and also yields higher quality libraries in the end.
Wow, that's fantastic. Where did the idea of creating Twist Biopharma come from?
Yeah, so the idea of creating Twist Biopharma came from our founders. So Emily, who's my boss, CEO of the company, as well as some of the other founders on the executive team, came up with the idea of creating the 2 verticals I talked about. So Twist Biopharma, as well as the data storage verticals. And so they then sought out people to come in internally to kick off those new verticals. And so, and again, the idea was really this idea of not just selling custom DNA products, but also selling the application of them. And so that's why we see the Twist Biopharma, as well as even data storage, as kind of the next evolution of creating even higher value products derived from DNA.
I know we haven't, it's a GPCR focused podcast, but can you tell us a little bit more about the storage, the other division of Twist?
Yeah, again, it's not my expertise, but it's just this idea that you can use DNA for long-term data storage. So as you know, DNA is one of the most rugged materials that nature ever created. So in the fact that for example, you can dig up, fossils and collect their DNA and actually understand like what organism they came from. So the idea is that rather than encoding data in kind of ones and zeros on silicon, can you actually encode for data in the A, T, Gs, and Cs of DNA, and then take advantage of that rugged nature of DNA, and so synthesize it, and then if you need to down the road, actually read it out. But it's more of a longer term storage rather than kind of like short term read-write storage that you might think about in terms of a hard drive.
Wow, truly revolutionary, definitely.
Yeah, super cool.
It does sound really cool. So as I mentioned, it's a GPCR focused podcast. Where do GPCRs come into play in the Twist Biopharma's mission?
Again, I always say synthetic antibody phage display libraries are actually best used for really difficult to drug targets, because if you had an easy target, you might want to just kind of use the more traditional antibody discovery technologies like hybridoma or all the other kind of more mainstream technologies. But and if you think about it, like what are the most difficult targets out there that are difficult to address with biologics? Certainly GPCRs are first and foremost on everyone's mind. So And also, as we know, almost a good majority of the validated targets out there are directed against GPCRs, but they're mostly drugged by small molecules. So again, we had this idea of, again, applying it to a class of targets that are difficult to drug, but use synthetic antibody phage display libraries to address them. And so that's where this idea of targeting GPCRs came from. And we wanted to take advantage of our unique way of generating libraries using alga pools to create a library that was kind of unprecedented in the industry in terms of addressing this specific class of targets.
And the goal is to be able to generate antibodies targeting any GPCR that we know at least the structure for, or is it just, or how do you start out? What's the starting material in order to be able to generate an antibody?
Yeah, so our very first library came from this idea that you could encode for specific GPCR-binding motifs into the heavy chain CDR3 of an antibody structure. And so we created a fully human synthetic antibody phage display library where we basically collected together all the known motifs that are known to bind to GPCRs. So motifs derived from protein ligands, protein peptide ligands, maybe even in some cases, even antibodies that are known to bind to GPCRs. And we created, again, a very large alga pool that encoded for all these different motifs and then inserted it into the top of the heavy chain CDR3 of this library. So I call it a motif-directed library. And it's basically rich in things that bind to GPCRs. And so, ideally, what we found is it's very straightforward to find antibodies where some of the motifs derived to those specific GPCRs are in the library. And we actually have a really nice example where we've done that with GLP-1 receptor. But we also find that it can also be used to drug other targets that maybe don't bind proteinaceous ligands, or even in some cases that maybe are orphan GPCR receptors. So even though this motif content is directed to specific GPCRs, we can find in the context of larger antibody diversities in the other loops, we can actually find antibodies to some of these other GPCRs that fall into these other classes of either non-proteinaceous GPCR binders or orphan GPCRs.
And it's a nice way also to think about GPCR families. So having an antibody that targets one specific receptor in a family, but having that proximity in the sequence and the topography of the receptors that is exposed at the cell surface can also be an advantage of using this technology.
Right, exactly.
So how does a platform work? Let's say you want to target a family of receptors. What do you do in practice? So how does it look?
So like I mentioned, we have these off-the-shelf libraries that we've already created. I talked about the motif-directed one. We've also created a GPCR library that's where we've basically taken all known GPCR-binding antibodies, and we've actually taken all those sequences and found the commonalities between all of them. And we've also created another library that's focused on GPCRs. So we have those two libraries that we can use to just kind of pull off the shelf and pan against any GPCR we wish. We just need to have a good source of the actual GPCR target, which often is a high expressing cell line that we can get our hands on, or in many cases, we can work with others to get to solubilized GPCRs that we can use to pan and screen the library against. And so once we have those two things, we have all the expertise and resources in-house to rapidly pan, screen. And then at the end of the process, we can also very easily synthesize all the DNAs that encode for all these antibodies. And we've also created a whole workflow for making antibodies and doing all the downstream testing to see if the antibodies bind cells and actually also have function against these specific GPCRs. So we've streamlined the entire workflow. Again, starting from these high quality libraries, as well as having access to high quality target that allows us to very quickly find antibodies that bind, as well as potentially have function against each of these different GPCR targets.
Wow. And when you say very quickly, what is the time frame that you usually look for? And what are some of the regular, old school type of ways of coming up with GPCRs? How does that compare with the technology that you're using?
Yeah, we've streamlined it and added as much automation as we could to potentially get it down to like an 8 to 10 week period. And we've shown that from time and time again, as long as we have access to the off-the-shelf libraries, as well as have access to these cell lines, we can very quickly and rapidly progress to that. I've set up the workflow such that we can kind of skip kind of the more traditional fragment-based screening and go directly to full IgG. And so that allows us to give us a bivalent molecule that is very amenable to doing a lot of the assays that are typically done with GPCRs in terms of looking at flow cytometry binding, as well as doing all the functional assays. So that'll really rapidly allows us to get to antibody materials that we can then test in these assays to see if we have potent GPCR antibodies.
It's really neat because in less than 3 months, you go from a sequence to an antibody that you're able to test for binding, but also test for functional response.
Yeah, absolutely. So I think that's also a really big game changer for what we do, is this ability to not only have access to these really rich sources of libraries, but also there's a speed at which we can implement those workflows to get to an antibody that's in the past been really difficult to get, because GPCR antibodies are just so intractable oftentimes.
Yeah, and they're difficult also to obtain if you go the old school way or the longer
absolutely.
route.
I mean, again, I often love to look for, I call my greenfield areas where there's not a lot of competition, because there's just really no technology that enables you to drug specific targets. And so that's why I love these GPCR libraries, because I can go after targets where others have tried really, really hard and they've oftentimes failed. And so I can go in with these libraries and really allow me to drug them quite readily and potentially easily compared to some of those previous efforts.
So this would be one of the major distinguishing characteristics of Twist Biopharma and the GPCR platform, that you can generate antibodies really quickly, and then you can also test them in-house to look for binding and for functionality. What are the other characteristics that distinguish Twist Biopharma from a traditional, or this methodology from a traditional way of looking for antibodies?
Sure. The other major distinguisher that I haven't talked about yet is just our ability to quickly make lots of additional libraries. So I call it my library of libraries. This idea that in the past, to make a single synthetic library, maybe it would have taken maybe a year to do in a traditional company. Here, I can actually make new libraries on the order of on a monthly basis. And so we continue to increase the breadth of our library of libraries by adding additional library content every month. And my vision is that eventually we'll get to maybe even having like a whole plate of different libraries. In phage display, you're oftentimes capped out around 10 billion different antibodies per library. And so one way around that limited diversity is to actually have multiple libraries. And so if I have 100 libraries at 10 billion different antibodies per library, then I could roughly have 10 to the 12th different antibodies in total that I can screen. And so that's one way where, again, because we're experts in DNA, we can very quickly make these high quality libraries and make lots of them and have them all at our fingertips, where if any target comes our way, we can pan not just one, but maybe multiple libraries all in tandem using this high throughput workflow that I talked about before.
That's great. Who do you usually collaborate with? Who are these libraries intended for? Who is Twist Biopharma marketing its services for?
Yeah, again, we're here to serve the entire biotech and pharma industry. I think initially, a lot of our partners have been smaller companies that don't have access to a discovery group internally. So they see us as a value-added partner to help them in their antibody discovery efforts. But now we're transitioning and trying to work with larger companies, pharma companies, to not only do that work for them, but also to license out the technologies to them, because they now of course have internal groups that can interrogate and use these libraries. So we're also trying to actively work with larger companies to out-license our phage display libraries to them as well.
Wow. And what is the best way to get in touch with you if let's say I'm a biotech or I'm a pharma company, I have an interest in subclass of GPCRs, what do I do?
Yeah, we have our own website, twistbiopharma.com. You can just go on the website, and there's a contact us tab you can put in. Of course, you can always contact me over LinkedIn as well. That's also a great way to get in touch, and we can set up some time to go through in detail all the different libraries that are in the library of libraries.
That's fantastic. You seem to have, the way I imagine it is kind of this shelf where you have all these treasure libraries, and you kind of, it's like a CD shelf, and you can pick and choose.
Yeah, exactly.
Antibodies that you wanna use. So one other question I had for you is, so now you're making libraries of libraries. What is the plan? Where do you see the company going in 1 to 5 years when it comes to libraries and developing this technology or using it better?
Yeah. So again, as I mentioned, there's really kind of two kind of buckets of libraries that we plan to do. So I think we definitely plan to create more kind of general use libraries that are based on different scaffolds. So Fab, SCFV, VHH, single domain libraries, which also have a lot of applications in GPCRs. To just have them on the shelf where you can again interrogate difficult to drug targets, maybe that aren't integral membrane proteins, but just again any target that might come our way, we can use these general libraries. And then we also plan to continue this trend of finding libraries to difficult to drug targets. So again, to expand on our GPCR efforts, to think about other targets like ion channels, transporters, carbohydrates as targets. And again, continue this theme of creating target-focused libraries at the same time. So we see both types of libraries as being really important, and that again will allow us to continue to expand the breadth of the library of libraries.
Of course, and having more and more partnerships with biotech and pharma will also, my guess is, I mean, you'll say what you think about this, but would also allow you to kind of direct where the need is and try to respond to where the need is in biotech and pharma.
Yeah, absolutely. And I mean, another big area for us is we continue to collaborate really closely with other companies too. Like we're working with academics, other biotech companies to come together and create libraries together. And so that's also another way that we see how we can continue to innovate and create even more libraries is by working with others that may have unique expertise in particular types of antibodies or have some unique kind of bioinformatics and silico expertise. And so we're very open to collaborating with others to come together and use this fantastic DNA platform that we have to create these very innovative libraries.
Super. What are some of the applications in patients, for example, where you see these antibodies do what they're meant to be doing, meaning targeting GPCRs? Where do you see those applications happen? Would it be more drug discovery, or would it be directly treating some diseases? What are your thoughts on that?
Again, we're not really focused internally on coming up with our own drugs quite yet. We're kind of more outward facing when it comes to using these libraries. And so again, we're very focused on seeing what our partners are interested in using them for. I mean, most of the targets we've seen lately are mostly oncology focused, much like most typical biologics out there. But certainly I think in the area of GPCRs, there's a huge breadth of targets in other diseases. And so I'm sure we'll see other applications in inflammation, cardiovascular disease, maybe even some other disease areas that'll probably diabetes, for example, that might play a role in other metabolic diseases as well.
Yeah, well, GPCRs do basically everything when you think about it.
Yeah, no, it's a huge breadth of different different diseases you could get involved in. So I think that's again really a really nice thing about working with GPCRs is you can really work on lots of different applications in terms of diseases and potentially have a big impact, not just in oncology, but in all these other really very relevant and kind of unmet diseases that people are trying to drug.
Yeah, and having such a cool platform and all these really, really innovative tools that Twist Biopharma has developed, I think could be considered a great foundation to build on and to move ahead drug discovery, and then also understanding how these GPCRs function in the future so that we can target them better.
No, sure.
That's fantastic.
Absolutely.
So I have to ask, how have you been doing with the current pandemic? I know everybody's talking about it, and we cannot not talk about COVID. How has it shaped the company's work? How has it affected the team? Can you tell us a little bit more about that?
Sure. So again, Twist has been really fortunate in the sense that we've been able to keep the R&D and manufacturing part of our company going during the pandemic. So that's been good for us to help our partners, a lot of who had to shut down during this time. And so we were able to keep their projects going, which was fantastic. We also undertook a COVID project of our own within the Twist Biopharma team. As you may have seen, we've done some discovery work against both the virus as well as ACE2 to come up with antibodies that could be used for either therapeutics or diagnostics. So doing those projects as well as having the COVID specific project, I think was actually really helpful to keep the team motivated. We were actively working on something that could help out with the pandemic, and at the same time delivering to our partners all the fantastic work that was not COVID related, but was certainly important to them to keep going during the pandemic. And so I think by having those two types of projects going through the team was really helpful to keep us motivated and pushing ahead through these really difficult times.
And focused as well at the same time. I think it's a good motivator.
Absolutely.
And you know that you're directly working on this whole COVID.
No, totally.
Were there any changes within the company that you had to implement to adapt to the new reality? How did that happen in the company?
Yeah, again, we had to adhere to all the typical safety precautions that everyone else is implementing in companies in terms of masks and social distancing, and also, as I mentioned, the current at our headquarters, the only people that are here are really the folks that are doing lab related work. All the other office staff are staying home. So I think being really focused on using the site to do the really important research work and manufacturing work that we have to do, and then keeping all the other stuff away from that site as we move ahead has been really important for us to make sure we stay safe, but at the same time keep things moving ahead.
Yeah, and it also helped adhere to all the social distancing rules.
Yeah, absolutely.
At one spot, definitely. Well, Aaron, thank you so much for being here today. We've learned a great deal about the Twist Biopharma platform. I can't wait to see what's coming. I do still really imagine that freezer with all the libraries coming together, and hopefully now people in the GPCR field will hear more about the work that you do, and they can get in touch with you through LinkedIn. And on the podcast transcript page, they'll also find the link to the company, but also the link to your LinkedIn account. Great. Thank you so much, Amina. And I really look forward to interacting with anybody that wants to get in touch with us around any GPCR efforts they're doing. As I've publicly said, I love working on GPCR targets. And so anybody that needs help in their discovery efforts, please get in touch with us. Absolutely. And especially that you have the capacity of generating a lot of antibodies very quickly, but also you do have the capacity and the expertise to test these antibodies, which is unique. Absolutely. Yeah. No, totally. All right. Thank you for being
Thank you so
here.
much.
Thank you.
All right. Bye.
Bye.
Thank you for listening to this Dr. GPCR podcast episode. We hope you enjoyed it. Thank you to our sponsor, Twist Biopharma, for the support. I'd also like to thank Dr. Aaron Sato, Attila Forrest, Jin Chong, Shivani Sachdev. Music by Rosa Bershish. I'm your host, Dr. Yamina Bershish. To support Dr. GPCR, please visit drgpcr.com/sponsors. We truly appreciate your support, which allows us to bring you podcast episodes, newsletters, and so much more. Thank you for the privilege of your time, and until next time, stay safe.